Now let's study asexual reproduction and its types. Binary fission, budding, regeneration, fragmentation, spore formation, and vegetative propagation. Binary fission. It is a simple process in which a cell divides and forms two individuals. Amoeba is the most well-known example of binary fission. As you can see, initially the pseudopodia are retrieved. The body of amoeba is coiled and becomes round. Amitosis is observed. The division of the nucleus takes place, which are followed by the splitting of the cytoplasm. At the point of fission in the body of amoeba, a constriction starts to develop. The constriction or furrow turns deeper, resulting in formation of two daughter cells. Binary fission is further classified in three types. These are simple, longitudinal, and transverse. An example of simple binary fission is amoeba. For longitudinal fission, we have Euglena, and the examples of transverse binary fission include diatoms and paramecium. Budding. This is observed mainly in Hydra and yeast. In the structure of Hydra, there is a bud that detaches from the main stem and forms a new individual. The most prominent difference between binary fission and budding is that in binary fission, the parental identity is lost. That is, there is no difference between the parent and the daughter cells. Also, in binary fission, the cytoplasm divides equally, whereas in budding, parental identity is not lost because there is an unequal division of cytoplasm. Fragmentation. It occurs by the breaking of parent body into two or more parts, each of which is capable of growing and living independently. For example, algae like Spirogyra and bryophytes such as Marchantia and Regia. Regeneration. Now in this type of asexual reproduction, the organism breaks into parts, but each part is not completely sufficient to live as a new individual. Thus, first the missing parts of the organism are repaired by the proliferation of cells, and then it lives off independently. For example, Hydra, Planaria, and sponges. Spore formation or sporulation. Members of the kingdom fungi and simple plants like algae reproduce by spore formation. Special asexual reproductive structures are formed in these organisms. Zoospores. These spores are made inside the cavity, because of which it is known as endogenous motile spores. They are biflagellated spores and are mostly found in algae and fungi. Sporangiospores. Found in fungi, they are also formed inside the cavity, so they are also known as endogenous. They are non-motile because they lack flagella. Conidia. These are the most important type of spores and are also found in fungi. The stalk of their structure is known as conidiophore and the extensions are known as steric. Above this are formed Conidia, which are young at the base and mature at the tip. Hence, it has a basipetal arrangement. These lack flagella and are also non-motile. They are exogenous in origin because Conidia are formed outside the cavity. Gemule Formation. This is mainly found in sponges, which produce a tough-coated dormant cluster of embryonic cells for development in more favorable conditions. Vegetative Reproduction. In this type of reproduction, a new part is not formed, rather an old part of the plant is used to grow a new individual. It is of two types, natural and artificial. Natural vegetative reproduction is also known as vegetative propagation. It can occur by reproductive roots such as in dahlia and sweet potato, a leaf in Bryophyllum and Kalanchoe, a stem with underground modification such as tuber in potato, a corm in Colocasia, a bulb in onion, garlic and lily, a rhizome in ginger, lotus, turmeric and banana. Or we can also have sub-aerial modifications such as runner in oxalis and grasses, an offset in Eichhornia and pistia, a stolon in mint and jasmine, a sucker in pineapple, chrysanthemum, banana and mint, or a bulbil, which is a fleshy bud as in agave. Artificial vegetative reproduction. It is a type of plant reproduction that involves human intervention. The most common types of artificial vegetative reproduction techniques include cutting, layering, grafting, suckering, and tissue culturing. These methods are employed by many farmers and horticulturists to produce healthier crops with more desirable qualities. Cutting. A part of a plant, typically a stem or leaf, is cut off and planted. Adventitious roots develop from the cuttings and form a new plant. Cuttings are sometimes treated with hormones before being planted to induce root development. Grafting. Grafting is a process in which two types of plants merge together to form a single individual. From one plant, we take a shoot system, and from another, we take a root system. The tissue systems of cutting become grafted into or integrated with the tissue systems of the base plant over time. Since the cambial tissue is really important for grafting, the monocots cannot undergo grafting. Only dicots can undergo grafting. In grafting, the shoot part is known as scion or a section, and the bottom part is known as stalk. Layering. This method involves bending plant branches or stems so that they touch the ground. The portions of branches or stems in contact with the ground are then covered with soil. Adventitious roots or roots that extend from structures other than the plant roots develop in the parts covered by the soil. Micropropogation. Also known as plant tissue culture, it is a method of propagating a large number of plants from a single plant in a short time under laboratory conditions. This method allows propagation of rare and endangered species that may be difficult to grow under natural conditions and are economically important or in demand as disease-free plants.